So we've been talking about feedback loops, and I want to continue talking about feedback loops, and in particular talk about the implications of feedback loops for a very, very important idea in biology, which is called the Central Dogma of molecular biology. So what is that? Well, shortly after Watson and Crick in the late 40s and early 50s worked out the helical structure of DNA, Watson announced what they called the Central Dogma of molecular biology. And it's kind of taught, unfortunately, in a lot of classes straight from that today. So the central dogma is pretty simple to state. It says that DNA is the ruler and that instructions go from DNA to RNA, and that once transcribed, the instructions go from RNA to protein, and there is a unidirectional flow of information from DNA to RNA to protein. And that's what is generally called, as I picked this off the net, but you could pick 100 more like it. These are all illustrations of the central dogma, DNA to RNA to protein. The problem with this is that, number one, it is completely wrong. It is absolutely disproven. There are well-known counterexamples, which I want to talk about. And what's even worse is it's not just bad science, which it definitely is. It has given rise to some really bad social implications of science. It has given rise to racist science. It has given rise to some really bad science because of its obsession with genes and with the idea that genes are everything. And that isn't true. And it's produced a lot of bad science and a lot of bad social theory because people have not taken account of the incredibly important role of feedback relations. So the first blow to the doctrine of the central dogma of molecular biology was delivered by a very interesting person named Barbara McClintock. You probably never heard of her. So she got her PhD from Cornell in plant genetics in the 30s and 40s. And she started working with maize, you know, Indian corn, the kernels of the corn that have all the different colored little kernels. All of those color patterns are genetically determined. And so she was an experimentalist, and she took a big plot of land, ruled it with string into one square, little squares, and in this square, she crossed this with this. And in this square, she crossed this with the square of the cross of the this with that. And she did all of these experiments, and she spent years analyzing the data. Her analysis of the data led her inescapably to the conclusion that there were occasions on which RNA reprogrammed DNA. And when the Watson and Crick types were told that, they said, that's impossible, cannot happen. I just told you, the central dogma of molecular biology is DNA to RNA to protein. As one biologist told Barbara McClintock at the time, it's like the father to the mother to the children. There is a strict order of command. And McClintock stuck to it, and she said, no, this is what my data are telling me. And they told her again, they pulled out another metaphor. And a molecular biologist said to her, you know what it's like, Barbara? It's the boss and the secretary and the workers in the factory. And the boss dictates the letter, and the secretary types the letter, that's transcription. Secretary types the letter, and then takes the letter to the workers in the factory who make the protein according to the instructions of the boss as typed by the secretary. Now, Barbara, we know that there can be bad secretaries. Viruses can mistranslate the DNA. But Barbara, the secretary does not reprogram the boss. There can be mistyped letters, but the boss himself is never altered. The DNA is immutable. And she said, no, my data say there are mutable elements in DNA and that the RNA in the right circumstances can push them around. So her work got published, as you see. She published in 1950 in PNAS. But the work was totally ignored. 20 years later, in 1975, or before 1975, Howard Tiemann and another group of molecular biologists, this was now a subject, found that certain viruses could carry out what they called reverse transcription. Now, you know what transcription is. It's the path from DNA to RNA. And reverse transcription is the path from RNA to DNA. And in particular, reverse transcription may ring a bell with you if you've studied HIV at all. You know that the way HIV takes over the cell machinery when it infects you is with a protein called HIV reverse transcriptase, which is how HIV, which is an RNA virus, alters your DNA by the process of reverse transcription. So everyone said, oh, this is a fantastic discovery, this Nobel Prize. And then someone said, you know, there was a crazy lady, that crazy lady at Cold Spring Harbor, Barbara McClintock, said this 20 years ago. So they looked it up, and sure enough, she had said it 20 years ago. And so she shared in the Nobel Prize that was given to Tiemann and the others for discovering this process by which RNA reprograms DNA. So that is just a counterexample to the central dogma. And by the way, I have to say something. I approached one of our esteemed biology instructors who was teaching the central dogma, and I said, hey, John, not his real name, I said, hey, John, you really have to stop saying that because it isn't true. And take, for example, McClintock and Tiemann and the Nobel Prize for reverse transcriptase. And he said to me, he said, yeah, the central dogma is not 100% correct. It's a simplification that we have to teach at this level. Well, I don't believe that. I don't believe it's ever right to teach stuff that's just wrong. And the central dogma is just wrong. So let's go on to look at some more cases in which it turned out to be wrong. Again, shortly after Watson and Crick, some very good French biologists, Monod, Jacob, started to ask a question. For example, how do we get repeated expression of a gene, except by making the gene work again and again and again? Is there some feedback present? And they said, oh, yes. And this was the work, incidentally, that led to the lac operon, which we have just been discussing. They said genes have responsive elements that are not coding for proteins, but are rather the receptacle for protein action. So the gene makes a protein, and the protein comes back and binds to the responsive element. In the specific case they were talking about, it was the positive feedback that constitutes the action of the lac operon. And so this positive feedback loop, they pointed out, can turn on expression for a long time after a single stimulus. And that's a great property to be able to have. And what they showed was that this positive feedback mechanism, which was from DNA to protein back to DNA, is a very important feedback loop that produces important phenomena in the production of this protein. Jim Farrell, who is at Stanford, wrote a very nice paper recently, or more recently, in which, and I want you to look at the title, Self-Perpetuating States in Signal Transduction. Positive feedback, double negative feedback, and bistability. So this is all stuff that is beginning to ring a bell. Bistability is something we've been talking about. And what Farrell points out in this article is exactly what we've been saying, which is there are all sorts of system properties that you want from a gene regulation network, like self-perpetuating states, like a permanent effect from a transient stimulus. And how does the system do that? So as Farrell said, talking about Monod and Jacob, they drew diagrams. They weren't writing differential equations, but they were drawing diagrams that represented positive feedback loops. And these positive feedback loops included the lac operon. And this was the work for which they won the Nobel Prize. And they were able to show that this positive feedback loop here was embodied in the E. coli cell, and by means of this sigmoidal positive feedback here, produced this biological switch. So the question that they addressed is, how do you make if given the central dogma, how do you make a biological switch? And the answer is, you can't. And so therefore, to have a biological switch, we must be incorporating feedback mechanisms that turn things on and off. And as you know, and this is now just repetition, we've talked about this, we have in this system the three equilibrium points of the bistable system and the existence of a biological switch. And so saddle node bifurcation, which is what we talked about in the context of the lac operon, means that in this region, for example, we have a stable, an unstable, and a stable equilibrium point, and a robust biological switch which can turn on expression. What this says, and this is kind of a big picture here, is this is an interesting kind of math, this feedback loop, bistability, the other examples of equilibrium points and bifurcations that we have been talking about. This is a math that talks about qualitative changes. And I'm just going to say qualitative change equals bifurcation. And in each of the systems that we looked at, there is a qualitative change that occurs. You remember the deer-moose system? For the mild competition parameters, we had coexistence of the two species. That was a stable equilibrium. But if we just increase that interspecies competition term by a little bit, the system underwent a qualitative change. The coexistence equilibrium point is unstable. You now get two new stable, which is all deer, no moose, all moose, no deer. But if I ask you the question, why does the deer-moose system change from coexistence to no coexistence, there's an answer. And the answer is the bifurcation theory answer, which is that the reason why that change happened was because the intraspecies competition parameter passed a critical point. We saw another example of qualitative change in the spruce budworm model. The spruce budworm sat at a low population. Then when there was a little bit more carrying capacity, a little bit more leaf space, the birth rate went up a little bit. All of a sudden, you had a qualitative change to a new equilibrium point, which was the outbreak equilibrium point. And so the bifurcation theory is the answer to the question, why does the spruce budworm have outbreaks? Answer, because R and K wandered into the bistable critical region. Why does the lac operon switch turn on? Answer, because that R, that degradation parameter, passed a critical point. Now all of a sudden you have a switch. We saw that example of political opinion. It was balanced at the center. Then as that bandwagon effect got larger and larger, all of a sudden the center equilibrium, which had been stable, is now unstable. Two new stable equilibria, all N or all P or all left and all right or all whatever you like. Why did that polarization occur? Because the bandwagon effect strength passed a critical point. So the take-home point here is that this is math that we're doing, but it's interesting in that it has qualitative implications. People think of math as quantitative. Mathematicians calculate numbers to 4 degrees of accuracy. No. This is a math that explains qualitative changes. So with that in mind, I want to go on and show you some more examples of the use of bifurcation theory to explain qualitative changes in biology. In particular, every time in biology we see a concept of a biological switch, we should immediately suspect a bifurcation as producing that switch-like behavior. And I want to talk about two concepts that are used in biology a lot. They're used as words. They have scientific definitions. The concept of differentiation and the concept of maturation. And I'm going to show you work done by others modeling these two phenomena as saddle node bifurcations. So our first example of a saddle node bifurcation is going to be the definition of the concept of differentiation. So what is differentiation? Well, you can have, at the ultimate, you can have stem cells. They can advance a little bit and become pluripotent. Pluripotent means that they can be committed to a lot of different cell types. They can become bone cells or they can become neurons. And that process by which a pre-cell becomes committed to become a neuron, or a pluripotent cell becomes committed to be a neuron or a bone cell, that is differentiation. And in a very interesting paper that was in Science about 5 years ago, Mary Teruel is a professor at Stanford in the very interesting Department of Chemical and Systems Biology. And her paper demonstrates, as she says, that differentiation is a cell decision switch. The cell decides to become committed, and what are the factors that produce that? So she has this nice diagram. So adipocytes are fat cells. And she's dealing with a pre-adipocyte, that is a pluripotent cell that can commit to being an adipocyte or not. And the stimulus that makes the pre-adipocyte commit to the adipocyte lineage, a very interesting drug called rosiglitazone, which is one of our major anti-diabetes drugs. So in the presence of rosiglitazone, the pre-adipocyte becomes committed to the adipocyte lineage. Now look at what she says. These pre-adipocytes differentiate through a bistable switch mechanism with a single threshold for activation that involves positive feedback between two key transcription factors. And then she has her flow chart and a true differential equation. And you'll notice that here is the positive feedback loop. P par gamma enhances CEBPA, and CEBPA feeds back positively onto P par gamma in the presence of the differentiation stimulus. Her two-variable differential equation has sigmoidal activation right there, which you're totally familiar with. She's using a cube to denote a slightly steeper sigmoidal activation than the square from the lac operon. But this is a positive feedback loop, and it's a positive feedback loop modeled by a differential equation. So she studies that differential equation and produces, guess what? A saddle node bifurcation diagram, which you completely recognize. Here's the unstable, here's the stable and the stable. In this case, the low stable is the pre-commitment adipocyte. The presence of the stimulant rosiglitazone moves you to the right in this diagram and eventually moves you to the point where there is no longer an equilibrium point there, and you have to jump up to the other available equilibrium point, which is committed adipocyte. And so this is a classic bistable switch, almost identical to the lac operon. So the basic idea of her paper is exactly what we are saying. Here is a saddle node bifurcation producing a qualitative change, namely from pre-commitment to commitment. Let's look at another example. Interestingly, also from Stanford and also from the same Department of Chemical and Systems Biology. Jim Farrell is a very senior, highly respected researcher. He was the author of that paper we were just talking about before, about persistent states and the work of Jacob and Monod. Here's an interesting paper where he is talking about maturation. So what is maturation? Well, you have an oocyte, and before the oocyte can be fertilized, it has to undergo a qualitative change from just being an oocyte to being a mature oocyte. And that is a qualitative change. The chemical that brings about that qualitative change of maturation is progesterone. And so in the presence of progesterone, you go from immature oocyte to mature oocyte. So Farrell is actually able, just like in the other paper, Farrell is able to state what the positive feedback loop is. He knows the names of the players. He knows that P42 MAP kinase feeds back positively onto MOS, and that is a positive feedback loop. And then he draws the diagram of the activation-inactivation, and this should look pretty familiar to you. The only difference between this diagram and our Lac-Operon diagram is that in our Lac-Operon diagram, the sigmoid was fixed and the straight line moved. Now, this is a slightly different case. The straight line is fixed and the sigmoid moves up and down. But you see that the geometric effect is exactly the same. This sigmoid crosses the blue line once, right there, has a single steady state. As that red curve comes down, as the sigmoid comes down, as you see, a bifurcation occurs, and now we have 1, 2, 3 equilibrium points. They are, of course, a stable, unstable, stable, as he correctly labels. We now have the biological switch that constitutes maturation. Immature is down here, and mature is the high steady state. He draws a very clear saddle node bifurcation and labels it for you. So the increasing progesterone, just as always, the increasing progesterone, you stay at the low state, stay at the low state, stay at the low state until it reaches a point where there is no stable equilibrium down there anymore, and now you have to go up to the other equilibrium branch, which is the mature state. And he says, the process of oocyte maturation is viewed as the traversal of a saddle node bifurcation. He concludes with an interesting point, which I want to stress for a second. So first of all, he says oocyte maturation arises from a saddle node bifurcation. That's great. Look at the last sentence. This type of reductionistic systems biology holds great promise for understanding complicated biochemical processes in simpler terms. I think that's a heck of a statement, and I think it's very, very, very true. I think, I'll be honest, I think we get buried in the biochemical details. I think we get buried in the molecular biological details, and we memorize lots and lots and lots of details, and we lose the big picture. And the big picture, as he correctly says, the big picture is we would like to have a switch that turns on maturation, and here's how to arrange activation and inactivation to pull off that kind of phenomenon. And it is, I believe, and I think he does too, really important that we begin to look at biology in these higher level terms. So we can go back to the central dogma, which is the green at the center, and now we add McClintock's feedback loop, and we add the Monod, Changeau, Jacob should be added there, all of those folks with protein to DNA positive feedback loops, and that is, of course, in their case, a positive feedback loop. So there were two major feedback loops that invalidate the central dogma of molecular biology. Just for completeness, because you already saw, Farrell already referred to this. This is another major feedback loop, and we have not discussed it yet, and it's going to be our next topic. So this is a sort of introduction to the next chapter. Here's a very pretty paper that was in Science 15 years ago. HES1 is a super important notch binding protein, which is very important in laying out the body plan in embryogenesis. And these folks looked at it and they saw a very interesting thing, which is that it oscillates over time. Gene expression oscillates with a 2-hour period, and you will notice that it's going up and down by a factor of 4. Now, in a lot of labs, if you have gene expression went up by a factor of 4, you start writing the paper. That's considered a major increase in gene expression. But what these folks are saying is that happens every 2 hours on its own. And this is an oscillation in gene expression. So what could possibly cause an oscillatory gene expression? We have been talking about a bistable gene expression produced by a positive feedback loop. What produces oscillatory responses? And the short answer is a negative feedback loop. And that's going to be our next topic. And by way of introduction, this is the abstract from their paper. And you will notice that they're saying, first of all, it oscillates with a 2-hour cycle, and it is caused by negative autoregulation, or to put it another way, a negative feedback loop. That's going to be something that we are going to be talking about a lot. Here is their model. There's another major case of oscillatory gene expression, which we will also be talking about, which is the gene P53. Very interesting paper from the group at Systems Biology at Harvard Medical School. They took this gene and they took another product which is activated by the gene and then feeds back negatively on the gene. And they just watched it over time. And what they saw, just like shark tuna, the two oscillated with the green, P53, always leading the pink. The green leads the pink, the green leads the pink. It's like the green is the tuna and the pink is the sharks. And that's not a bad metaphor here. And what they found was, when they looked at it over time, a 2 to 3 hour period, green, red, was clearly seen as a pattern of gene expression. In fact, negative feedback and oscillations were the subject of the Nobel Prize in 2017. Hall, Rosbash, and Young, these are their words. Notice that they say oscillations are achieved by delaying various steps in the network. And what they show is that these two genes produce gene products which come back and inhibit the genes that were producing the products. And what we're going to learn in the next unit is that that kind of negative feedback, especially in the presence of time delays, that negative feedback can produce an oscillatory response. And in this case, the oscillatory response that is produced is the circadian rhythm, the 24-hour rhythm of the body. So we will talk about that as we begin to talk about oscillation. But for now, I want to leave this picture in your head. And I hope you stare at this and replace the central dogma with this better approximation, in which DNA goes to RNA, goes to protein, but then protein has positive feedback on DNA, RNA has reverse transcriptase on DNA, and protein feeds back onto DNA. And this is, of course, a negative feedback loop. So in the presence of these feedback loops, now, obviously, it doesn't really matter which way we hold the page. There's no chain of command from DNA to RNA to protein because protein is giving orders to DNA.